we're looking at a map of Philadelphia for a reason. Okay, this is where I live. I'm not trying to show you where I live, but what I'm displaying here on the map, you can see if we zoom in, you'll see that there are houses, there are buildings, you can move around. And what you can see here is I'm displaying a search for the word coffee. So I'm displaying all these coffee shops in this particular geographic area. So each of the entities on this map, almost each of them has a GPS location. Okay, so that's how the map knows where they are. The GPS location is just a coordinate, right? It's a coordinate on the space, which is the map, basically. So if you didn't get to that in the queries, we had a search called a collocation search, right? And I'll show you what that's doing in a second. But basically on a map, if I have the coordinates of all the coffee shops and I know the coordinates of all the apartments in the region, right? I can say, well, let's say I'm moving to Philadelphia and a very important thing for me is that I need a coffee shop, 10 meters from my doorstep. Because every morning I need to go get my coffee. And if I don't get it, then I cannot function, right? Then I can make that a requirement. I can say, find me all apartments that are within 10 meters of a coffee shop. Okay, and then it'll return me a list of apartments. So and the way it's doing that is by virtue of the coordinates, the GPS coordinates. The location of the apartment is known, the location of the coffee shop is known, and their location is on the same space, the map, so I can co-locate them. Okay, so in this context, we use something very similar in a genome space. So we have a genome, this is represented by this line here. The genome has genes on it, which have locations. We know exactly where they are. We have their GPS coordinates. And we have SNPs, and we know where they are. We have their GPS locations, right? Their GPS coordinates. And you can do this for any entity that can be placed on the genome map, basically. It'll have a coordinate. And by that, you'll be able to actually then use this concept of collocation, collocating them. So I can say, well, I want to find all the genes that are within 10, instead of 10 meters, I'm saying 10 base pairs of a SNP, for example. Okay. And so there were a couple of queries in the previous exercise that asked you to follow up the search after you found your SNPs, is to collocate them with the genes. And if we go to the one search I wanted to display is the search for the artemisinin resistance gene, the CALS13. And this one is a bit of a tough one, right? So it asks you to define the SNPs from this particular chip, right? And you follow the parameters in the exercise, and in the end you come up with something like 12 SNPs. And then the question was, well, are any of these SNPs anywhere close to CALS13, right? That's the gene that's involved in artemisinin resistance. And so, but these are SNPs, of course, and we want to somehow collocate them with genes. And so the way you do this is you add a step, and now you use a genomic collocation. So this is my GPS collocation that I'm doing on the map. And now I can look for genes based on taxonomy, because I want to find all the genes from Plasmodium falciparum. In this case, I'm going to pick 3D7. Oh, sorry, actually, I'm doing something wrong here. So you can do it for all the genes, but let me go back. I was mixing up queries. In my case, I didn't want to look at by taxonomy. I'm looking for a specific gene, right? The CALS13 gene in this case. I want to collocate it with a specific gene. So instead of the taxonomy, I want to run a text search because I want to look for the word CALS13. In my case, I know that it's the name of the gene is K13. So I'm going to look for K13 or it has K13 at least in the name. And I'm going to specifically limit this to the product description. You don't have, there are many ways of getting to the same end. I could have typed CALS13 and left everything selected, and you would get just a larger number of genes, and then you may have to limit your results after that. And so I run my search, the collocation search, and this is basically my GPS configuration tool, right? So I can say I want to return all the genes. So instead of apartments, I'm returning genes whose exact location in this case overlaps the SNPs. So instead of coffee shops, I'm looking for SNPs. So I'm saying I want to return all the genes that are exactly overlapping with my SNPs. And if I run this search, the result, I ran this before, and I think other people ran it too, but the result is 0, right? So none of the SNPs, none of these 12 SNPs overlapped with the CALCH13 gene. So then the question is, well, maybe the one of those SNPs is close within a certain distance of the CALCH13 gene. This is where it becomes tricky because, well, how close, right? And the exercise, I don't think, gives you any clues as to how close you should go. So it becomes almost like a trial and error, and it could be on either side of the gene. So now instead of saying, I want to return genes whose exact region, I'm going to say I want to return genes whose custom range. I'm going to customize the range here. And I'm going to say, well, genes where we can start with minus, let's say 5,000 upstream and plus 5,000 downstream, okay? So that means I'm looking for any SNP that is within this exact region surrounding the entire gene and the gene itself, okay? And I can run the search. And again, the answer is going to be 0, right? Which becomes frustrating, right? Because you keep trying these things. And so there's no magical answer here. The bottom line was that you were supposed to try this by trial and error and try different distances. It turns out if you go up to 7,000 base pairs, you will actually get CALCH13. So CALCH13 is within 7,000 base pairs of one of these SNPs here, okay? Now, does it mean that any of these SNPs are truly involved in artemisinin resistance? We don't know. All this is saying is that in this SNP chip that was used to define CALCH13 mutations, you can identify the ones that are within proximity or within a certain distance of, in the samples we have, that are within a certain distance of the CALCH13 gene. So remember, the search criteria to begin with, you looked for parasites that had slow clearance compared to fast clearance, right? And that's one of the hallmarks of CALCH13 of artemisinin resistance. And so basically, as you accumulate mutations that result in artemisinin resistance, you get slower clearance of the parasite. So they're surviving longer in the presence of artemisinin. And so in this case, you found 12 SNPs. It turns out that some of them are within 7,000 base pairs of the CALCH13 gene, which is known to be involved in artemisinin resistance. It's possible that many of the other genes that are in that same region are also involved in artemisinin resistance, potentially. And minimally, it tells you that this is potentially a hotspot for where there's some accumulated mutations that could be involved in artemisinin resistance. Okay, hopefully this was helpful, but this collocation tool is quite useful for getting genes that are within a certain distance of a SNP or returning the SNP. In the toxoplasma one, we ran the search. I don't know if I still have it here, but we ran a search where basically we identified a bunch of, I don't think I have it anymore, but we identified a bunch of SNPs, and well, yeah, I don't have it. So after we did that, the question was to collocate the SNPs with genes, but you could have also modified this and said, well, I want to find the genes where the SNPs are in the 500 nucleotides upstream of the gene, right? So maybe you're looking for SNPs that are potentially affecting the promoters, because not all the SNPs are going to be coding, and not all of them are going to be ones that affect the structure of the amino acid. Some of them are going to affect the expression of the gene, and that might actually have real big differences in resistance or whatever phenotype you're looking for.
Can I just add something?
Yes, of course.
So the other thing that is important with the artemisinin example, which I'd overlooked, so apologize to my group if I confused that, but just in case any other groups were thinking were having the same issue, it's important to realize that for that particular experiment, the SNPs came from a microarray, not from whole genome sequencing. And when you have SNPs from an array, you're basically only looking at a predefined set of SNPs. So it's entirely possible that the reason why we didn't find a SNP that's actually in the CALCH13 gene is that SNP is just not on the microarray, so we're never going to see it from this particular experiment. So one of the advantages of looking for SNPs in the local region is that you may find SNPs that are linkage disequilibrium, so segregating with SNP that's important. So we'll never actually see the SNP that is important, but we can get an idea of locations on the genome that are important, and from that maybe narrow things down a bit.
Yeah, good point. Thank you for that, Catherine. Yeah. Okay, so I think we're going to switch over now to our final group project or group presentations. So this is a 2-day project. We have one more formal structured exercise tomorrow morning. We'll do metabolic pathways just to give you a flavor of that. But the rest of the time, you're going to be working in your group on your group projects. And so, so to give you some idea what those group projects are, open this up. So as this picture or movie indicates here in the background, is that it's a group project. You know, all of you in each of the breakouts, you're going to be working together to put together a project. So that's going to be one of the key hallmarks of this, is that it needs to be collaborative between the people in the breakout sessions. There are goals for these projects. So as a group, you know, we'd like you to, in your groups, we'd like you to start discussing with each other some possible questions that you may want to use the databases to answer. Okay, and I'm going to give you an example of one question, but there are many questions that you can come up with. Over the last few days, we've explored the databases in many different ways. We've asked questions about genes just simply by searching with the text. We've asked questions about the expression levels, whether it's transcriptomics or proteomics level. We've done searches looking at orthology. We've looked at polymorphisms and many more, right? There are many things we've done over the last few days. One of the key things that we've done is that we've tethered these searches together in a search strategy, right? We are leveraging data from many different sources, and we're putting them together to try and build an in silico experiment. So even though some of the questions we were asking maybe were very simplistic, find me all the genes that are upregulated in oocysts compared to tachyzoids, for example. It's a straightforward question, but it becomes very powerful when you can combine the answers to that question with other questions that you may ask. For example, how many of them are potentially secreted? How many of them have at least 7 transmembrane domains? And how many of those that have 7 transmembrane domains and are upregulated in oocysts also have a high number of non-synonymous SNPs? And how many of those don't have orthologs in mammals, right? Right there, without even developing a question, there must be some interesting genes in that list of results, right? You could build an entire project on to try and answer some true biological questions in the lab. Another rule is that as a group, or another goal, as a group, you want to work together to develop. So after you come up with a question, the goal is to then use the database to develop a search strategy or search strategies that you are going to use to answer the question that your group put forth, right? And then finally, we want to hear from you about what you found out. And so tomorrow in the afternoon session, each group is going to present a maximum of 10 minutes, sort of a quick summary of what they did and what they found, okay? There are some rules, okay? So one of the first rules is, you know, the goal is to have fun, right? This is, all of you are scientists. We've all been, you know, together, you know, going through exercises over the last few days, and we've been focused on these exercises. But hopefully now you can start having some fun with the databases and with each other in terms of trying to answer some biological questions. This rule number 2 is that you need to come up with a really cool name for your group. So however you want to display that, but we will make a channel where you can put your group name in there. But that's a key thing that's been always fun in these group exercises. It's very important that you work collaboratively. So I know some people are more likely to be quiet in a group and others talk more, but I think this is a time for everybody to say something and contribute to the group thinking. As such, the instructors are not always going to be with you in the groups, right? We're going to hop around. And in many cases, we won't be talking at all. We'll just be listening in, but we'll be there to help you, to maybe guide you. You can bounce some ideas off of us, and we can help you with sort of channeling, maybe what kind of searches you can run. And the final rule is that each person in the group needs to speak during the presentation. So this is not going to be a single elected official from each group who's going to, give the presentation. Each person should give something, even if it's like a small intro. Like, we were interested in finding genes that are potential vaccine candidates in Plasmodium. Switch it over to somebody else who's going to describe what is our first search, right? And so don't stress over it. The idea is this shouldn't be a stressful event. We all know each other now. We've been together for multiple days in breakout sessions, and nobody's grading you on this, right? You're not going to get a grade, but we are going to learn. We are all going to learn from each other. So I'm going to hop over quickly to ToxoDB. And let's see, I think I've got here. Okay, there we go. So whichever database you end up asking your question in, I think one thing that's very useful is to look at this section here. And this always gives you an idea of what kind of data sets are potentially available, right? So if you're going to run a phenotype search, right, you can open it up and see, oh, look, in ToxoDB, there's CRISPR phenotype data, right? So now, all of a sudden you can start asking about the essentiality of genes. If you're, so I would totally recommend that you open all of these and look at what kind of searches are available. So if in your group, the decision is that, okay, we're going to work on toxoplasma and we're going to find XYZ in toxoplasma, then definitely spend some time looking in here and seeing if what kind of searches are available, because the worst thing you want to do is limit yourself to one type of search. And then after, multiple hours of work, you realize that, oh, there are 5 other searches that could have been very useful in answering your question and potentially taking you 10 steps ahead before you go to the lab to do your experiments. And so I'm going to just do an example. So let's say I'm trying to find some virulence genes, potential virulence genes in toxoplasma, right? And we know toxoplasma is an intercellular parasite that infects mammalian cells or any kind of nucleated cells for that matter. And we know that it sends out proteins to the host, and there are different kinds of proteins, but I'm going to limit myself and ask, well, does toxoplasma have any kinases? So I'm just going to start by searching for the word kinase in the database. Okay, so there are 13,000 or close to 13,000 genes. You see here in the filter on the left, genes in ToxoDB. These are not only toxoplasma genes, they're in ToxoDB that have the word kinase in them somewhere. So I'm going to now limit. I clicked on genes here. I limited to my genes. I have 12,942 genes. I'll export this to a strategy. Okay, so I have now all these potentially interesting genes. Okay, so another question I might ask here is, well, if these genes, if these kinases that are effector proteins or virulence factors, and they're going out into the host, it's possible that they're under pressure to change, right? They may have a higher number of non-synonymous mutations because they may get exposed to the immune system, and if they get taken out of the equation, then those parasites are potentially have a selective disadvantage. But the ones that maybe, if they vary these proteins, maybe they have active advantage. This is just a hypothesis, but I'm going to try it out. So I'm going to add a step and search for genes based on SNP characteristics. And I'm going to select all the data sets. So because let me change it to ME49 in this case. Select all the data sets. And I'm going to ask for non-synonymous SNPs, and let's make them 20. Again, this is an arbitrary number. I'm just basically, in my mind, I'm assuming that if a gene has at least not 20 non-synonymous SNPs, that's a lot. It's more than 10, it's more than 5, it's more than 0. So that means that there are some changes happening there. And I'm going to run this search. Okay. So as a result, I come up with 188 genes. Again, now we're down to Toxoplasma gondii MV49 that are potentially kinases and that are potentially under diversifying selection. They have a higher number of non-synonymous SNPs. So I wonder how many of these are predicted to be secreted, right? Because if these genes are virulence factors that are going into the host, right? So maybe they have a secretory signal peptide. And so let's add another step and ask exactly for that. So let's look for secreted. And so you come up with this search here for predicted signal peptide. Let me add the search. I'll select all, it doesn't matter because my intersect will only intersect with Toxoplasma gondii NV49 and run the search. So out of 21,000 proteins in the database, 17 of them were part of my 188. So of course, at some point you want to go down and look here, and you'll see here I have a column for this expression values from this one experiment. This may be interesting. You may want to look and see if these genes are changing in expression depending on which stage of the parasite they're in. But as you start scrolling down, you will notice that there are a number of genes that do make sense. So rope tree proteins, kinases, these are well-known effector proteins, microname proteins, but then there are other ones that are unknown, right? So here's a protein kinase putative, right? To me, this would be super exciting, right? I would want to go and if this gene is coming out with all these known virulence factors, I wonder what that kinase, that unknown kinase is doing. So I would be interested in exploring that. And of course, as I mentioned, you could continue. So I can say, well, I wonder if any of these genes, maybe one of these genes would be a really great vaccine candidate, right? So I'm going to make sure that the one I select to do my vaccine studies does not have any orthologs in mammals, because I definitely don't want to elicit a self-immune response. So I'm going to add a step here, and let's try the phyletic pattern search, right? What we were doing earlier today. So orthology phylogenetic profile, and I'm going to define any group that does not contain mammals. So under chordata, here's mammals. I'm going to click twice on this. So this search will define any group that does not contain any mammalian protein and intersect the results with my previous search, which is those 17 potentially secreted kinases. Okay, and so if it turns out 15 of them are also there. So these are 15 potential kinases that have at least 20 non-synonymous SNPs that are potentially secreted and do not have orthologs in mammals, right? And so, and you can keep going. I can add a step. We saw that there's phenotype data, right? And so I can ask, well, I wonder if any of these are truly essential for the parasite. So I'll make sure that, let's pick -4. I think that's a good number. So between -4 and -6.8. Here's a graph down here that shows you, dispensable genes all the way on the right and essential genes all the way on the left. Going from -6 point something all the way up to +2 something. And so that down here in this quadrant, below, and I can change this threshold. I chose -4, but maybe that's too stringent. But anyway, that gives you an idea. Oh, this is a good area. Maybe I will start off with, and then I will modify my search. And so let's run the search. And again, at some point, the answer may be 0, right? And it's just exactly what happened here. So none of those genes that I picked here are essential for fitness, at least in the experiment, in that particular assay that was run for the CRISPR. But again, I was very stringent. So let's revise this and make it a -1, for example. And again, I don't know what the answer is, but the cool thing about this is that, and they're still not there, which is fine. But that's the cool thing about this, is that this is an in silico experiment, and you really haven't wasted any reagents. But now at least that those are not essential based on this particular assay in the particular stage of the parasite, which is something you have to keep in mind as well. So you may choose to take this at face value, or you may choose to say, well, okay, I'll put this as a footnote somewhere, but still, I do think these 15 genes are quite interesting, and I may be interested in taking one of these back into the lab and working with it. Like there's a hypothetical gene that fits all the characteristics, but in theory, nobody knows anything about it, that I may want to actually explore further and study it more to see if there's Maybe there's something known about it, or maybe not, but it's a good start. Okay, and so in a short order, I wasn't asking bioinformatic questions, right? I was asking biological questions, and in fact, bioinformaticians, the goal is always to ask biological questions, but you're using bioinformatics to answer those questions, and so we're just using these tools here with the data that's available to construct a strategy that we constructed here, combining multiple different data types and analyses. And this is, I didn't even explore any of the transcriptomic data sets, right? I could have asked for stage specificity and all these things. So there's a lot more that I could add here to make this even more robust or more interesting. Okay, so this is all I wanted to say, basically, in terms of getting ready for the group exercises. Are there any questions from any of the participants, or anything any of the other instructors would like to add? So one of the key things is that in your group, as you're discussing the question you want to answer, make sure that you're all reach some kind of a agreement to the question, right? You don't want to be, one person saying, well, I'm going to look for drug targets in coccidioides, and the other person says, I want to look for, secreted kinases in Plasmodium, right? Let's try and agree on for each group, agree on one question or one theme that you want to answer. And then you can work, individually, but maybe one of you would like to always share a screen so you guys can talk together and try and construct a strategy. And so I highly recommend that. And for tomorrow, when you do your presentations, in a few weeks ago, I was in Catherine and Eve and myself and David were running a workshop on fungal genomics. And there, some of the students had elaborate, PowerPoint presentations, and others just displayed the website and showed the strategy and walked us through the strategy. And both were equally effective, right? So don't feel the pressure that you have to build a beautiful PowerPoint presentation. That's not the goal. The goal is really the search strategy and what you've done with it, okay? That's the most important aspect of this. So if there aren't any questions, I would say that we should go ahead and open up the rooms. The instructors will be floating around today. We'll bounce in and out of your rooms. We'll be there to help you answer questions. Use the Slack channel to chat with us, and make sure, if you have a question and there's no instructor in your room, send a SOS and tell us that you need the instructor in your room. Rachel, if we haven't done that yet, we should also set up channels for each group. So maybe we'll make, depending on how many groups we end up breaking up into, we can make group channels. And so each group can also have a place where you can chat with each other in the Slack workspace.
Sure, I can set those up. And it looks like we'll have 7 breakout rooms.
7? Okay.
7 groups?
So okay. Yes, so Rachel is going to put us into 7 groups, and this will be the same groups obviously tomorrow, because you discuss. So you'll have a couple of hours today to discuss this. Tomorrow morning, we're going to spend about an hour or so on metabolic pathways, but then you will still have a few more hours until around 1 p.m. before we do the group presentations. So you still have time tomorrow to work on this. And really, don't stress. This is not supposed to be an exam. You're going to really hopefully have some fun and come up with some cool ideas and use the databases to potentially come up with some projects that you would feel confident maybe going to the lab and researching, or some areas where you think more research needs to be done and more data types need to be incorporated to help improve the databases as well. All right. Okay. So Rachel, I think we're ready to go into our breakout rooms.